Hello, everyone. Welcome to a NanoString-sponsored LabRoots presentation for Neuroscience 2021. I will be the moderator for this session. My name is Elizabeth Schneider. I'm a Senior Global Marketing Manager here at NanoString. Today, we have the pleasure of speaking Dr. Frances Edwards. She's a professor of neurodegeneration in the Department of Neuroscience, Physiology and Pharmacology at University College London. And today she will be speaking about using the NanoString GeoMx digital spatial profiler to look at gene expression in microglia and astrocytes in relation to plaques. Dr. Edwards is a CNS synaptic physiologist. She graduated with a bachelor's degree in behavioral pharmacology from the University of Sydney in her native Australia, also had a master's degree there as well. She moved on to electrophysiology, gaining her PhD between the Australian National University and the Max Planck Institute for Biophysical Chemistry. And her current lab specializes in electrophysiology, but also adds on gene expression, immunohistochemistry, and molecular techniques. And a primary aim is to improve mouse models of Alzheimer's disease by investigating the role of different Alzheimer's disease risk factors in knock-in mice with Alzheimer's mutation. So thanks everyone for joining, and thank you, Dr. Edwards. We're looking forward to your presentation.
Thank you very much for that nice introduction. So we have just recently discovered the NanoString technology, and it's really, I think, brought up some very interesting new things that we can look at, so starting with genes that we sort of knew something about, but then bringing in both cell-specific and regional-specific information. And this makes a really very big difference to how we can interpret it. Alison van Schooik is the person at NanoString who's been incredibly helpful and has done a lot of has done all the sort of technical side of getting the data. And Jack Wood and Eugenia Wong are students in my lab who've done a lot of the processing of the data that I'll talk about today. And we are funded by Cure Alzheimer's Fund and Alzheimer's Research UK, and this is all done in collaboration with John Hardy as well. So we work on Alzheimer's disease. Just a very brief introduction. I think most people have some idea about Alzheimer's disease and its devastating effects on cognition. Basically, it starts with the deposition of extracellular plaques, which are ringed in circles in this tissue here. They're in the extracellular tissue, and they build up over decades before any diagnosis occurs. Sometime again, several years before the diagnosis is likely to occur, the tau tangles start to build up, and these are now the ones that are in the triangles here, and they're inside the neurons in the brain. And it's only once these tau tangles start to build up that you notice a massive loss of synapses and brain tissue, and you see on the right there the horrendous effect of Alzheimer's disease on the brain by the time the person eventually dies, often 5 or 10 years later. So that's a very great effect, but it takes years before you know that you actually have this disease building up. And there's no treatment at the moment that can slow or reverse the progress of this disease. There are some helpful treatments for some of the symptoms, and there has just recently been released a drug which mops up amyloid beta, so it'll be interesting to see how that goes. But this is it's something that needs a lot more information. So that's just the background of the sort of thing that we're looking at, and we're trying to understand how that disease progresses from amyloid beta plaques through to tau tangles and onwards. Another feature of the disease, which has become clear over the last few years, is that microglia and the innate immune system of the brain are very closely involved somehow in the progression of this disease. We showed quite a few years ago now in 2015 that there's a 0.97 correlation between a certain module of microglial gene expression and the buildup of amyloid plaques. I mean, that's the highest correlation I've ever seen anywhere, I think, in physiology. As well as that, in GWAS studies, it's become clear that the risk factor genes, the variants of genes that increase the risk of being diagnosed with Alzheimer's disease, are very often genes that are very highly expressed in microglia. So it seems clear that there's something very important about the interaction of microglia, plaques, and synapses and so forth. And that's what we have ended up working on now in various different ways. Just very briefly to introduce the nanostring technology. Of course, any questions about this should go to the people at nanostring rather than to me, who'll be able to explain in much better detail. This is from a post that they showed me when they first introduced the technique to me a little quite recently. So they have a proprietary reagents which label all the RNA in the tissue. So you have sections, very thin sections, which get labeled with these oligo-labeled antibodies. So they label all the RNA in your section, and then they're able to photo-cleave from those labels, effectively a barcode. So by cutting off the DNA oligo, they're able to collect those into a tube, and you can see the step-by-step method across on the right, where the section gets stained with these oligo-conjugated antibodies. They're also stained with antibodies to show what regions you want to be collecting from. So, for example, we have stained microglia or astrocytes with antibodies, and they're able then just to cleave the RNA that are in those areas of interest that you have labeled on the section. They then take those into a counter and can count the number of any particular, the number of barcodes that they have for each of the genes that are expressing in your section. So that's the sort of basic concept of the nanostrings, and I think it becomes the nanostring technology, and I think it becomes even clearer when you see what our experiment is. So the idea of the experiment is to assess the effect of the vicinity to the plaque on gene expression. So as I said, we've shown already that microglial genes are expressed, or that a bunch of microglial genes are expressed in incredibly close correlation with the buildup of plaques. So we want to know whether it's just that they're only expressed when they're near the plaque, or whether they're expressed generally in the tissue, and exactly how those genes interact with the plaque. We've mostly worked on microglia, but now we're starting to work a lot on astrocytes as well, another sort of immune cell in the brain. And so what we've done here is looked specifically at the gene expression in microglia or the gene expression in astrocytes, and we've looked compared to where they are sitting in relation to plaques. Now, I should say at this point that it's important to make clear that you're not actually doing single-cell RNA-seq effectively. You're not actually collecting just from microglia. It's going to be highly enriched for microglia. But what it's doing is you label the microglial cell, and then it cleaves in that place where that label is. So if anything is just below or just above, it will collect that RNA also. Extremely enriched for the cells because the sections are only 8 microns thick, and that's pretty much the thickness of a single cell. So they're very highly enriched for microglia, or very highly enriched for Alzheimer's, for astrocytes, in our study. So we're looking at different mouse models. We have wild-type mice, the ordinary mice. We have mice that are expressing the Trem2R47H mutation, which is a microglial gene, and this particular variant of the gene is a very strong risk factor for being diagnosed with Alzheimer's disease. Unfortunately, in mice, it also decreases the Trem2 expression somewhat, but so you could say that they are Trem2 incompetent, these mice. We then have APP knock-in mice. These are mice that have a mutation, have 2 mutations in their APP, and APP is the precursor for the amyloid beta that builds up and starts the whole plaque deposition. So those mice start to get plaques from about 9 months of age. It's not a very aggressive model, unlike some models where they get their plaques very early and get all sorts of phenotypes. These mice get their plaques only at about 9 months, and it continues gradually to express more and more, or to lay down more and more plaque as the mice get old. We consider this very important because aging, of course, is a major part of Alzheimer's disease. It is generally a disease of old age, and so we think it's very important to use truly aged mice. So the mice we're using here are 18 months, and we also use mice right up to 24 months of age. It's very common in Alzheimer's studies to see mice of 12 months being referred to as aged mice, but actually mice live for 2.5 even years, or even a little bit more than that. And so really a 12-month-old mouse is probably the equivalent of a 35-year-old or something of that sort. So we use 18 to 24-month-old mice largely, and particularly in this study, and so we think that this is truly aged mice. We then have mice where we have crossed the APP knock-in mice with the Trem2R47H knock-in mice, and so these we can then have a look at how this risk factor affects both the plaques and how they develop, what happens to the microglia and what their phenotype is, and indeed the astrocytes. And so this is the idea, is both to see what happens in terms of the plaques, but also to see how the Trem2R47H mutation affects this. This was just a trial study using the TAP program at NanoString. So this was just a very first trial. We're going to repeat this and increase the numbers. So what we had in the first instance was 3 wild-type mice, 3 Trem2 mice. So they're the mice that don't have plaques. And then we had 4 each of the plaque-bearing mice, the NLF and the NLF Trem2. So these, unfortunately, in the process, we lost the information from 2 of the mice. So we ended up with 2, 3, 3, 4, 3 as our sample size. A total of 14 mice, a total of 12 giving us information. This is quite a small sample size. We're very keen to increase this, but it is remarkable how much information we've already got even from this small sample size. Now, how the system works is that you have a slide like this. This is a standard sized slide. And you see that there's an area there marked out, 36.2 by 14.6 millimeters. And your sections have to fit within that area. How the analysis is costed when you're working with the TAP program, and it varies depending on how you're doing this, but when you're working with the NanoString's TAP program, how this works is that you pay per slide, and each slide comes with 12 regions of interest or areas of interest, and you can then pay for extra areas of interest in lots of 12. So you can, however, fit as many sections as you like on a slide, or as you can reasonably fit within that area of 36.2 by 14.6 millimeters. We managed to fit the 14 sections, so one section from each of those 14 mice, onto 2 slides. So we had one slide with 8, there's the schematic on the top of that, and one slide with 6 sections. And as you can see, we have a mixture of the genotypes on each of the slides to try and avoid any batch effects and so forth. On those slides, the slides then look like this basically. So this is a picture of the slide number 1 there with the 8 sections. You can see on the left-hand side the actual literal picture of the slide. And then we blow up one of those sections there in the middle to show you what it looks like. So the first step is that you get these, you make these 8-micron sections and you put them on a slide and you send those off to NanoString if you're doing it via the TAP technology. Of course, ideally we end up with a GMX machine at UCL and we can do all of this ourselves, but at the moment this is the situation. So we sent those off to Alison van Schooik, and she then did all the labeling from then on. So she then labeled those with the proprietary probes, as well as with the antibodies which are listed up there on the top left, GFAP for astrocytes, TMEM119 for the microglia, and she also labeled beta amyloid and used DAPI just to label the cell body so that you could see your way around the slice. So you can see it in the middle there and then blown up a little bit on the right, but it's even clearer here in the next slide, I think. So this is just where she's selected the regions of interest. So actually just briefly going back there, you can see on the whole section, out of that section, those little white areas are the regions of interest. And we did that together with Alison for every single one of those sections over Zoom, over starting at sort of 6 o'clock at night UK time and going through to about midnight. We selected the regions of interest on each of those slides. Now it's important to understand the difference between the region of interest and the area of interest. So these are the regions of interest, and the regions of interest that we wanted to have were things that were related to plaques and things that were not related to plaques. So you can see now on the next slide, the one labeled A, the plaques are the turquoise, and those that's completely full of plaques. There's plaques everywhere in that section. if you look closely, even where you can't really see the big plaques, there are little plaques all over the place there. So all the cells in that region are going to be very close to plaques. The sections labelled B don't have any plaques in them, so that's going to be well away from a plaque. So in our mice that have plaques, we had regions of interest that were A with plaques and B without plaques. We then chose our areas of interest, and our areas of interest were those that were labelled both with plaque and with microglia, so where the TMEM119 actually overlapped the beta amyloid antibody. So that's looking at microglia that are actually touching plaques. And then we also looked at plaques that the other plaques in the area, the other microglia that are in the area A. So in other words, we had microglia that were actually touching plaques and microglia that were near plaques but not actually touching plaques. And then we had microglia that were in one of the regions B. We then did the same with astrocytes. In the case of astrocytes, we didn't try and do the ones that were touching plaques. Astrocytes have very long processes, and they'll be labelled by the TMEM. And it just simply means that you wouldn't know whether they were really touching the plaque or whether you would likely to be getting RNA from an astrocyte that was really interacting with the plaque or not. It's a much more complicated structure. And so we used astrocytes that were near plaques and astrocytes that were far from plaques. In other words, any astrocyte that was in A or any astrocyte that was in B. So those were our areas of interest, 3 different sorts of area of interest for the TMEM119 and 2 different sorts for the GFAP. Now, an area of interest, it's important to make clear, is not just one selection. It is anything within A, for example, that where we had the TMEM overlapping the plaque would count as one area of interest for one section. In other words, it's what ends up going into one tube to be analysed. So it's not just one little spot, but rather what goes into one tube to be analysed. So it isn't specific to a single cell, although they are collecting the signal from single cells, from lots of single cells, and they are then pooled simply because there isn't enough signal in a single cell. So it's one area of interest is all the microglia that are touching plaques in A, or all the microglia that are not touching plaques in A, and so forth. So that was the experiment, to look at those 5 different areas of interest in animals that had plaques, and then in animals that didn't have plaques, we obviously only had the equivalent of far from plaque, and we did that in an equivalent area to A here, but of course not having plaques. And that way we could be sure that we weren't just talking about areas of the brain that expressed different levels of gene expression. This is all within the hippocampus, I might say. Just for interest's sake, so on the right we have the cells blown up a little bit more, so you can see how well you can see the astrocytes and the microglia, and the laser comes down and collects the data from just where you see the green signal or where you see the red signal. So they are really quite distinct from each other. On the left here, you see another version of our regions of interest, these funny looking shapes. And I just included this because Alison is extremely careful and goes to a lot of trouble to make this work. You can see in this particular section that we have quite a lot of blood vessels. You see there's the astrocytes right along the blood vessels there being labelled in green. And those, we weren't interested in collecting those. They're likely to have a very different function and a different gene expression from the general astrocytes that cluster around plaques and so forth. And so she was able to bring the region of interest right into a tiny little channel to jump across those regions, and so avoid collecting anything from those astrocytes, even though the region of interest itself spanned both sides of the blood vessel. So that was extremely helpful. And so that funny looking shape on the right is a single region of interest. And so we could collect everything from the microglia within that shape, even though, and it is actually continuous, even though it doesn't look continuous from here. Okay, so that's how we actually collected the data. That's the data we have. And then they go off and they send it to the counter, and about a week later, or maybe 2 weeks later, depending on the busyness of the system at the time, you end up getting back a big Excel sheet with all the TPMs for each of those regions of areas of interest, and you get the counts for all those areas of interest, and you get a whole big software package that you can use to deal with that data, or you can just take it from the Excel sheet and analyze it in any way that you want yourself. They're extremely helpful. They're very helpful in helping us to get around the software. It means that somebody who's not really a bioinformatician can do a lot of analysis on this data relatively easily. So just to show you one of the things that comes out of this software, so this is the principal components analysis, and you can see that here the red is the astrocytes, the blue and green are the microglia, and you can see there's considerable separation between the astrocytes and the microglia, which gave us confidence that that was really what we were collecting. Just to show you some of the data we got out of this, which I think is quite remarkable, we've done quite a lot of RNASeq, and of course that gives you a lot of information about a lot of genes, but it doesn't tell you which cells those genes are expressed in, and it doesn't tell you where they are. This is partly overcome by people doing single-cell RNA-seq. That at least gets down to the particular cell type, and indeed the expression in a single cell, and tells you what sorts of different cells there are there, but it doesn't tell you where they are in the tissue. There are other regional things, such as RNAscope, but that will only tell you about a few genes and not genome-wide, as we have here. So here are just a few of the examples of what we got out of this data. So here are some of the DAM genes. These are the disease-associated microglia that have been analyzed using single-cell, that have been brought out of studies using single-cell RNA-seq. So looking at some of those genes that have been related to disease-associated microglia, and here we are looking at regional analysis. So all the graphs I'm going to show you, so a lot of them are pretty similar. If you look at the Trem2 graph there on the top left, you'll see that the left-hand group of 3 bars with the black outline is the NLF mice, the APP knock-in mice, which have plaques. The next group, the grey ones, are the ones that have NLF with the Trem2 mutation. And then we have just the single columns for the wild-type mice and for the mice with the Trem2 mutation, but no plaques. So those 2 right-hand columns give you a bit of an idea of what the background is, what you would expect from background microglia where there are no plaques. In the set of 3 columns for the NLFs or the Trem2 mice, the left-hand, number 1, is always the plaque, the ones that are actually overlapping the plaque. Number 2 is the periplaque, and number 3 is the area that's away from the plaque. That's on the bottom of the slide there, if you're not following me well. So the left-hand column of 3 is the plaque, the second is the periplaque, and the third is the away from plaque. So you can see here, for example, in Trem2, it's quite remarkable. There's a strong relation to plaque, but it is only the plaque, the microglia that are actually touching the plaque, that are showing increased expression compared to the periplaque or the away from plaque, and that's completely prevented when you have the Trem2 mutation. And as you can see, the background level isn't very different from what you get in the wild type or the Trem2 mutant mice. It's important to note that the wild type is only n equals 2 here, so you often see quite a big error bar. You have to be a little bit cautious about interpreting the level of the wild type column. These others, TyroBP, is rather similar. It's not quite as completely blocked by the Trem2 mutation, but similarly really only up right at the plaque. ApoE is a bit different. It's a bit more sort of varied, a bit more gradually coming down, so it's up already in the periplaque area, and it increases further as you get closer to the plaque. It's blunted by the Trem2 mutation, but not so completely stopped. So these other ones, as you can see, have various different versions of that. There are a couple that are particularly interesting here. I think in terms of the let me have a look. Sorry, the CTSL1 I think is quite interesting. You can see it gradually goes down away from the plaque, but it probably is going down away from the plaque rather than up at the plaque, because it's going down well below the level of the wild type and the Trem2 mice. The FTH1 is ferritin heavy chain 1. That's an iron transporter, and you can see that in that case it's down close to a plaque and up away from a plaque, and that is in fact that it's at normal level when it's well away from a plaque, but it's decreased as it comes in close to a plaque, and we found that very interesting as well. It's no more decreased right at the plaque as is at the periplaque area, but when it gets anywhere near a plaque, this ferritin gets downregulated. This is just giving you a comparison to what you get in RNA-seq. So you can see most of these genes I'm showing here with the columns. This is all at 18 months from our RNA-seq data, and you can see there's no significant difference when we look at RNA-seq. Whereas all of those genes, if you remember from the previous slide, TyrBP, ApoE, and all of these dramatically affected if you're able to look at it in this regional and cell-specific way. This is showing Trem2 in a little bit more detail from our RNA-seq data. This is only wild type versus NLF, incidentally, in this data. We haven't done the Trem2 here. So this is only wild type versus NLF in all of these graphs. You can see we've done this from 2 months old all the way through to 24 months old. And Trem2 does eventually go up in the NLF mice fairly subtly. It goes up by about maybe 20% or so, whereas you saw that at the plaque, it's actually up by double. So presumably, this is because we are just watering down the data when we look at RNA-seq and the whole tissue. And if it's only being upregulated in a particular cell in a particular area, you simply won't see it with RNA-seq. So even though these are actually n equals, as you see, n equals 10 to 12 mice or 9 to 12 mice in these different groups, the RNA-seq data doesn't show any significantly difference in most of these genes at 18 months, whereas you get with only n equals 3 to 4 these spectacular differences regionally using the nanostring technology. You can also get volcano plots just to show you more typical things that you look at in gene expression analysis. Of course, in these particular data sets, we can compare a lot of different things. In this case, what I'm comparing is the actual plaque data to just the periplaque data, so not even the away from plaque. So it's just comparing the microglia that are actually touching plaques to the microglia that are very close to plaques but not actually touching them. And you can see on the left in the NLF mice, there are quite a lot of genes which above the horizontal line there means that they're significantly different statistically in adjusted p-values, and the vertical lines show you, the grey vertical line shows you a 50% increase, and the black vertical line shows you a doubling in gene expression. So there you have a lot of the normal suspects, the Trem2, the TyrBP, and so forth, which are coming out as highly expressed when the microglia is touching a plaque compared to the microglial cells that are neighbouring but not actually touching the plaque. You can see that there are also some genes that are up in the immediately neighbouring region compared to the plaque region, but not nearly as many. On the right-hand side, we have the very same sort of data, but this is when we have the NLF mice with the Trem2 mutation, and you can see that nearly all of that upregulation at the plaque is lost. And down at the bottom there, you can hardly see the writing probably, but those little red, that little red writing down the bottom is all those genes that were so strongly upregulated in the NLF mice are way down there with almost no difference, and certainly no statistically significant difference between them. So the Trem2 mutation is completely preventing this very plaque-specific upregulation of all of these genes. We also looked at astrocytes. For the sake of time, I'm not going to go into a lot about the astrocytes, but we're getting some very interesting data there. So this is just plaque and away for the NLF mice, and you can see that near the plaque there are quite a lot of genes upregulated there also. Some of those are perhaps surprising genes that we thought were microglial, and you start to worry that perhaps you are getting microglial signal there. But actually, when you look at the literature, you find that quite a lot of these genes are quite upregulated, especially in aged mice. So, for example, let's look at the C1qa there. When you look at the Ben Barres database, which is this brainRNAseq.org, he quotes the mouse astrocyte aging data from Clark et al., and you can see that at 2 years in the hippocampus, the C1qa is well and truly upregulated. Counts are still low, but it is a lot more than it is earlier in development. And if you look at the full data for the C1qa, this is what it looks like, and you can see that the it's almost doubled in the near to plaque region compared to the far from plaque region. And that was rather different from what we saw in the microglia, which were largely right at the touching plaque area that it was upregulated. And again, Trem2 completely stops this change. And you can see that the far from plaque area is very, very similar to what you get in wild types in Trem2 mice. So this is another case where an interesting piece of information comes out that we weren't expecting once you do things in a cell-specific and regional-specific way. Just an area, a little level of caution. I have kept saying that they are cell-enriched rather than absolutely single cell. This is an interesting result, I think. In the microglial data, we get this gene MAP2, which is very clearly a neuronal gene. It's not a microglial gene. And however, where we are taking our sections, there are a lot of dendrites. It's a very, very full of synapses. And the microglia and astrocytes are in around the dendrites and the synapses. And so as dendrites are such fine structures, they probably do run in and around the microglia. And you're able here to see the effect on MAP2 of being close to a plaque and away from a plaque in this way. And so it's quite a dramatic difference there, where you are really strongly downregulated the MAP2 expression, possibly because you've actually lost synapses there. MAP2 is expressed actually out in the dendrites, so it's an unusual one to pick up. You know, most of the RNA will be picked up in the cell body, whereas MAP2 is expressed out in synapses. So this very likely reflects a loss of synapses right in and around the plaque. So it'll be very interesting to do similar experiments, labeling the neurons instead of labeling the microglia or the astrocytes. Just finally, for those who are gene expression people, just to say that the software from NanoStrings actually has quite a lot of other sorts of things that you can do with them. I'm no expert in this, but I just thought I would add that there's quite a lot of other different types of analysis that are in their software package that you then have access to when you when you have done this gene expression analysis. So I think this is going to be a very powerful technique. I'm very excited about it. We certainly are applying for money to do a lot more. We are doubling the data set for this particular analysis, and I think very soon we'll have really some very quite definitive data, both about these DAM genes, which it's not so surprising that there's interesting data for them, but some other very interesting things are also coming out of these data sets. Okay, thank you very much.